study how cells respond to changes in their environment, specifically changes in the availability of essential nutrients, and by altering their growth rate, cell shape, and composition. For example, the Levin lab asks how cells know when they are starving for carbon and how they reduce growth rate and daughter cell production to compensate. Research in the Levin lab will also hopefully help us understand how human pathogens are so adept at acquiring antibiotic resistance, a growing crisis in today's world. Dr. Levin, the screen is yours.
Thank you. Let me start my PowerPoint. Wait. Sorry, it's a little getting used to trying to do two things at once is difficult, obviously. Okay. Yay. Okay. So it's a pleasure to be here, and I'm happy to be able to share some of our work. I was really excited to go to Cambridge, but I'm also really excited to take part in this experiment. I'm going to tell you today about our work on how the environment shapes the cell. And I want to thank Pete and Mike and Hannah for making this possible. And I also want to proudly show off my Christine Liu art, which just says scientist. I wear it every day. So bacteria are tiny chameleons that adapt to suit their environment. They are able to change very easily in order to grow and divide, which is their dream in life. As the environment changes, so do they. So microbiologists, on the other hand, we are creatures of habit. We prefer our medium pre-warmed and pH 7. So many of us grow bacteria in lysogeny broth, or LB. Sometimes people use Muller-Hinton broth. It's actually the standard for testing antibiotic sensitivity is Muller-Hinton broth. Again, beef extract, casein hydrolysate, starch, it was just put together because bacteria grow in it. Sometimes people use M9 medium, which is a defined medium. And again, it was sort of just put together because people could get bacteria to grow in it. At the same time, though, even though these media work really well, sometimes it's useful to think like our model organisms. I love this cartoon, which says, day 37, I finally earned E. coli's trust. And I'm hoping to earn E. coli's trust, but if I were E. coli, I wouldn't trust me or anyone in my lab because we do some pretty terrible things to E. coli every day. For E. coli, though, in my lab, we think about LB kind of like a sunshiny day. I always like to think of E. coli, and when they get into LB glucose, at least at the beginning of the growth curve, they're kind of skipping and dancing through a beautiful field and growing very happily. However, the real world for E. coli is not LB. It's more like sometimes what happens in the Midwest in the spring, and we get a nice tornado touching down and sirens going off. And even pathogens, although we tend to think of them kind of trying to find a good niche in the human body, often their situation is not so great, especially for pathogens like Staphylococcus or E. coli that might find themselves in different parts of the human body. So E. coli, right, it likes to hang out in the colon, but unfortunately for us, at least, it sometimes finds itself in the urine where pH can range from 4 to 8 and nutrient conditions can vary tremendously. We know from a lot of work from Elio Schechter, starting with Elio Schechter and Ola Mola in the 1950s, that the environment is actually one of the biggest, has one of the biggest impacts on bacterial physiology, growth rate, size, and other things. So I love this image from over here, this graph, which is from this 1958 paper by Schechter, And really, it's a very simple experiment. They grew salmonella in 30 different media, which must have been super fun to make up. But and they generated different growth rates. So each of these little dots is a different growth rate. You can ignore this dashed line, but each of these points is a different media that generates a different growth rate. And when they looked at the bacteria, they noticed that those that were cultured in a nutrient poor condition and a poor carbon source were much smaller and grew much more slowly than cells that were cultured in nutrient-rich medium and growing with a shorter mass doubling time much faster. And this is just an image that we took in my lab to show that difference. So over here you have LB glucose. The E. coli are wider and longer than their same cells. So there's no genetic difference between these guys. These are cultured in minimal succinate, which is a carbon source E. coli really doesn't like, where the doubling time is closer to 80 to 90 minutes. And there's a threefold difference in size between these two organisms, between these two culture conditions, same organism. So we understand a good deal about how model bacteria respond to acute stress. If you go to a conference, a bacterial physiology conference or stress response conference, they'll talk about salt stress or heat stress or acid stress. And that's usually stress that occurs over a short period of time, say about 10 minutes to an hour, maybe 2 hours. We know a lot less about how non-model organisms respond to acute stress, though. How does chlamydia respond to acute stress? How does multi-drug resistant staphylococcus respond to acute stress? In a similar way, we really understand a lot about the short-term response to stress, but we understand a lot less about bacterial physiology under steady state conditions. So instead of just looking after 2 or 3 generations, looking after 30, 40 generations under those stressful conditions when cells have really adapted to that new condition and are really growing under those conditions. So again, I want to emphasize that the environment influences almost everything about bacteria, how they grow, how they look, how they respond to antimicrobials. And in my lab, we're really trying to understand that. And so Lizzie Muller, who's a 5th-year graduate student in my lab, who's going to be defending next month by Zoom, so I invite all of you to come watch her defend, came to my lab and was really interested in one environmental variable, pH. And she was interested in pH because pH is one of the things that is changing constantly in the environment. And it's also important for bacteria because all the proteins on their surface are constantly exposed to changes in pH. So in acidic medium, the pH of the periplasm for E. coli or the outside of the cell for a gram-positive bacteria is essentially equivalent to the pH of the environment. And so in acidic medium, the periplasm or outside proteins on the cell surface are exposed to acidic conditions. And in basic medium, those same proteins are exposed to basic conditions. This contrasts with proteins in the cytoplasm, which is buffered and tightly controlled to maintain a more or less neutral pH. So pH influences how bacteria grow. So this is the first little story from Lizzie. Which goes back to some of these proteins on the surface of the cell. So most of the proteins required to synthesize E. coli's cell wall are located on the cell surface. So again, this is just E. coli. We've got a cytoplasm, we've got the plasma membrane, and then the outer membrane, and then we have this region in between, which is called the periplasm. And in the periplasm is this thin cell wall. And that cell wall is made of essentially 2 major components. NAG, NAM, which are these sugars, which are linked together, and then these little peptide chains. And there are 2 major classes of enzymes, glycosyltransferases, which polymerize these sugars together, and then transpeptidases, which cross-link these peptides together, the peptide chains. And what's important is all of these crosslinks are what generates the bacterial cell wall, the peptidoglycan, and the peptidoglycan is you can think of it like one giant cross-linked molecule, but it's absolutely essential for the bacteria under normal conditions in the environment. So just a little bit of extra information. These peptides that are cross-linked by these transpeptidase enzymes, they're the target of a major class of antibiotics called beta-lactams. And I always like beta-lactams, not only because, they are awesome drugs, but also because they're one of the few chemical structures I can easily spot because they have this nice square ring structure right here. Because transpeptidases bind to beta-lactams, they're called penicillin binding proteins, or PBPs. So you're going to see a lot of PBPs in a minute, and I just wanted to give you that little piece of information. Okay, so Lizzie wondered, though, how does cell wall synthesis remain robust across environments? You have these proteins, they're on the surface of the cell, they're being exposed to acidic pH, basic pH, and they have to do this essential process. And obviously, it's very difficult for a protein to do that. How can these proteins carry out this essential process when pH changes so frequently? The answer actually is actually interesting. I think very interesting. So if you just look at the reactions, and this is true not only in E. coli, but in most bacteria, in the proteins that carry out the formation of these little subunits of cell wall, 2 sugars in this peptide stem, there are 12 reactions and about 14 enzymes in E. coli. In the outside of the cell, for E. coli, there are 9 reactions and over 38 enzymes that do this reaction. And these enzymes, there's a high degree of redundancy. Depending on the group of enzymes, you can knock out 1, 2, 3, 4, multiples of these. And at pH 7 and LB, you really don't see much difference in growth. So Lizzie wondered, maybe these enzymes, maybe redundancy is just another word for overlapping functions or activities that ensure robust growth across environmental conditions. Maybe this redundancy is not really redundancy. It just means at pH 7, maybe their activities are overlapping, but at low pH, like you might find in an organism or in the bladder or in the soil, or at high pH, maybe only one enzyme is actually active under the kind of edges of that pH range. So to really try and identify enzymes that are affected at different pHs, she went through all 34 loss of function mutations in 34 of these so-called redundant enzymes and look for growth defects at the low pH and high pH. So again, lots of PBPs. But what's important is all these enzymes in purple, and there are 34 of them, can be knocked out in LB at least singly, and we don't see any difference in growth rate in E. coli. There's only 4 essential enzymes in LB at pH 7, PBP3 and 2, which are transpeptidases, and ROD-A and FTSW, which are 2 glycosyltransferases. Everything else you can knock out, make at least single mutations in at pH 7, and the cells are perfectly happy. At least to our appearance, to us, they appear perfectly happy. There are 2, what she found though in this screen was that 2 of these 2 bifunctional proteins, PBP1A and PBP1B, are required at opposing pH values. So PBP1A and B, they have both, they're bifunctional. They have glycosyltransferase activity and transpeptidase activity. They're conserved across bacteria that have a peptidoglycan cell wall. And the only thing that was really known about them was that they were synthetic lethal at pH 7. You can knock out 1A or you can knock out 1B, but you can't knock both of them out at pH 7. So what Lizzie found was really actually pretty cool. She found that cells defective in PBP1B lyse at low pH. So when she sifts cells from pH 6.9 to pH 4.5, she saw that they wild type cells, not much happened. So you can just watch them grow. I could watch them grow. It's just really fun to watch bacteria grow, I think. But it's very sad for the PBP1B cells because in contrast to the wild type, what you're going to see is that they're going to grow and then they're going to start to lyse. And I'm just going to play that again because it's pretty dramatic. I feel like there should be sad music accompanying it, but I wasn't that swift with putting together this talk. But you can see these guys, they just kind of blow up. And this little guy, he develops a big bubble of membrane and just kind of sadly sits there. 1A, she saw something similar, maybe slightly less dramatic. Again, the wild type cells, so cells defective in 1A, wild type cells, when you shift them from neutral pH to high pH, they grow actually quite well. When she shifted cells defective in PBP1A to pH 8, so high pH, she saw them grow for a little bit, but then a lot of them develop these sort of blebs and some of them lice, and it just gets kind of messy. They don't all die like the PBP1B nulls at acidic pH, but there's a lot of death. So the conclusion of these set of experiments were that PBP1A and PBP1B have overlapping function. So under acidic conditions, PBP1B is the primary enzyme that's active, and under under basic conditions, PBP1A becomes more important. And at pH 7, their activity ranges actually happen to overlap, which is why you can knock out 1B or 1A, but not both at pH 7. And actually, we have some nice biochemistry that I'm not going to show you from Valdemar Vollmer and Alex Egan that indicates that actually PBP1A transpeptidase activity is completely gone at low pH, which is why 1B becomes essential at that condition. So pH influences how bacteria grow, how bacteria look, and how bacteria respond to antimicrobials. So one of the things Lizzie noticed by looking very carefully at cells cultured at low pH was that they were shorter. So these are just measuring cell length. pH 5.5, you can even see in these pictures, these cells are shorter, and they're just wild type E. coli, than at pH 8. And actually the difference in size is almost twofold. And the difference in size for cells cultured under nutrient poor conditions and nutrient rich conditions is threefold, and these guys are twofold. So this is actually a really significant difference in size. So again, these guys are short at low pH and longer at high pH. So when Lizzie investigated, she found that it's actually a problem in recruitment of the very last step in cell division. So in E. coli, division is triggered when one protein called FTSN is recruited to the septum where the cells are going to divide. And that seems to somehow lock the division machinery in some kind of on position, and then the cells are able to divide. So recruitment of FTSN to the septum appears to be enhanced at low pH. So cells at low pH have a higher concentration of FTSN. Just if you're looking across, you can see this is an FTSN GFP fusion. You see a higher concentration here at low pH than you do at high pH. So more cells have a very distinct FTSN ring at low pH than at high pH. And when she looked and just counted the number of cells where she could clearly see a ring indicating that their FTSN had been effectively recruited, that was clear. At low pH, she saw a much higher frequency of cells with an FTSN ring than she did at low pH. And this was inversely correlated with cell length. So when you get FTSN, the cells divide. So they're dividing at a shorter length than they are at high pH because fewer of those cells, it just takes, they have to grow longer to get more FTSN at that site. So again, FTSN recruitment is enhanced at low pH. So the third part of what Lizzie discovered that pH impacts is that it really has an outsized impact on how E. coli responds to antimicrobials. So when she did tests, she found that drugs that target PBP2, which is important for making cells grow longer, and PBP3, and which is important for cells that make the cross wall that allows them to separate, those drugs were much less effective at low pH. Mesalamine, which it was 32-fold, it needed 32 times more mesalamine to stop the growth of cells at pH 4.5 than it did at pH at higher pH. It was a little bit over here, These guys are all 8-fold, requiring more imipenem, cephalexin, azetrianum, and piperacillin at low pH than a higher pH. Whereas a drug that didn't target the cell division machinery or the cell wall synthesis machinery, chloramphenicol, it really wasn't that effective. Maybe even its activity was enhanced at low pH. So this is easy to see if you were looking at cell morphology. So mesalinum and imipenem, they target PBP2 in the elongosome. So when you inhibit the elongosome activity, the cells just get round because they can't coordinate cell wall to grow this way. They just kind of blow up into little bubbles. The PBP3 and the cell division machinery are targeted by cephalexin, azetrianum, and piperacillin. And when you inhibit that, the cells filament because they get longer, but they can't divide into shorter cells. And then cefzolodin, which targets these bifunctional guys, they seem to have a problem with coordinating cell wall growth evenly across, and you end up with these sort of lollipop-like structures. Okay, so at acidic pH, when you add mesalinum, you see something different than what you see at pH 7. So at pH 7, you add mesalinum, which inhibits PBP2 in the elongosome. And you can see at 0.1 microgram per mil, these cells are very round. And at 0.4, you don't get any growth. So if you shift pH to 5, which really doesn't impact the growth of wild type E. coli much in the absence of drug, you can see at pH 5, at 0.1 microgram per mil, you see slight rounding maybe, but not much, and you get growth at 0.4 nanograms per mil, whereas at pH 7, you get no growth. And the divisome cephalexin, which is actually a drug that's used in the clinic, these guys, cephalexin at pH 7, you see filamentation at 6.25 micrograms per mil, and at 25 micrograms per mil, you don't get any growth. At pH 5.5, really at 6.25, you really see pretty good septation and division. These cells look almost like exactly like they do down here. Whereas at 25, they grow, even though you do get some elongation, as if you finally kind of overcome whatever was going on at pH that protected it. But again, you can see that the length is mostly shorter. So what was also really cool is she looked at Staphylococcus, so obviously the firmicutes and the gamma proteos are more distant than we are as human beings from Saccharomyces cerevisiae, so highly diverged. But when she treated Staphylococcus at low pH with imipenem, which targets PBP1, which is a homolog of PBP3, the division protein that was protected in the previous slide in E. coli, she sees an increase in the MIC, so these cells become resistant to imipenem at low pH. What's actually kind of cool is that she sees the opposite effect for other drugs targeting other different penicillin binding proteins of staph. Okay, so I think the take-home message of all of this is that pH can impact enzyme activity clearly, and activity of these big complexes that make the cell wall. How they do this, we're not sure, and we think it might be different for different groups of proteins. One way is by changing surface charge. So obviously the isoelectric focusing point of enzymes of proteins changes with pH, and obviously on the outside, and the cell and the protein sitting on the outside of the cell, the surface charge is going to change with pH. So that might change their activity, access to their active site. It might also change protein-protein interactions. Lizzie has good evidence that recruitment of FTSN and activation of this big complex that actually is required to make the division septum might be different at different pHs, and maybe actually is recruiting different components. That might also explain some of the antibiotic intrinsic changes in intrinsic antibiotic resistance that we're seeing. One possibility is that there's a change in expression or stability, and in none of the 3 cases that I've just shown you have we seen that. So we really think it's changing how proteins interact, and or their surface charge and the activity of their active site. And if anyone's ever done protein purification, you know you're very careful what buffer you want to use. And we think that's basically what's happening, only these proteins aren't purified, they're just on the surface of E. coli. So the take-home message is a single variable pH can profoundly impact bacterial physiology without altering growth, and that understanding physiology in host relevant conditions may inform antibiotic use and development. It may be that depending on the site of infection, we might be able to modify pH, for example, and enhance the activity of an antibiotic that isn't working so well. And that's actually, I think, really exciting. And as somebody who's done basic research my whole career, the idea that maybe this might be useful in the clinic is really kind of gratifying. I want to acknowledge first E. coli. Without E. coli, we couldn't have obviously done these experiments. I was trained as a bacillus genetics and bacillus cell biology in Rich Lozick and Alan Grossman's lab. I've come to terms with E. coli. It does offer some very nice things, but obviously we've tortured it a lot here. I want to thank NIGMS for our funding. We have a lot of collaborators, some whose work, Valdemar's in particular, I've shown you. We have a nice project also going with Rohit Papu's group, Casey Long's group, and Ana Flores Morales at Notre Dame. And I also obviously want to thank the lab. It's a fun place to work, and we're always looking really closely at things and discovering new things. So, and I also just again want to thank the organizers for giving me this opportunity. Thank you.
Oh, thank you very much, Dr. Levin. That was fantastic. I appreciate the time you've taken to be here and share your research with us. Now, I think we have time for just a couple of questions here. So let me look on over and see what we got. So, ready?
Yep.
Let's do this. On the basis of, for example, pKa or some more sophisticated metric thereof alone, that PB1A, B are differentially active? I think that's the question. On the basis of something like pKa, are they differentially active? The question, yeah, so that's the question.
Okay, so we have not seen anything obvious on those proteins. They are part of big complexes, so it's possible that some of their interactions with other proteins and patches that are not obvious on their paraplasmic domains are changing. Another possibility that we've seen with some other enzymes that Lizzie's working on, these enzymes that are important for separating the cell into individual, dividing cells instead of sticking together with lots of peptidoglycan. They need these enzymes called amidases that separate them. It seems like at least one of them at different pH may have a change in a helix that kind of gets in the way of its active site. And so that provides a clearer mechanism. But with 1A and 1B, in vitro, we can see again with 1A, it's not active at low pH, but what we can't tell is exactly why that might be.
All right, thanks, doctor. Got another one for you here. Could PBPAB have antagonistic effects against each other rather than a redundant, quote unquote, same function?
That is a really interesting question. I don't think the biochemistry that is out there supports it, although I'm not sure there's one that says that's not the case. I think if it were antagonistic, I'm not sure why deleting 1B would be inactive at low pH if it was an antagonistic function, because then 1A you would think would be able to take over.
All right. And we have time for one more. Hi, Dr. Levin. I was wondering if you could speculate as to whether there is a function to bacterial cells being longer at higher pHs. Would this have been selected for by evolution?
Okay, that's a really exciting question, and Lizzie has tried to get good data on that. We actually haven't seen, I don't know what her data on high pH is, but she has experiments that sometimes reproduce and sometimes do not, and we're not quite sure why, saying that short cells, and again, not only we have a whole collection of mutants that are short but otherwise grow really well. You wouldn't know in a normal growth curve that they have any problem. Those short cells, depending on the day, seem to be resistant to pH shock. So really low pH, like pH 3, which would normally kill E. coli, or at least really kind of stop it in its tracks, and it's very unhappy. the shorter mutants that we have, and they're about 20% shorter kind of mutants, those guys seem to sometimes, depending on the day again, do better. So there might be an advantage to being short, which as somebody who's 5'2", really I appreciate, but I'm not sure. We don't, unfortunately, the data on that is kind of not as solid as I'd like it to be.
As someone who is 6'1", I can tell you there are definite advantages of being more diminutive, especially on airplanes.
Yes, I've heard that. So.
Thank you, Dr. Levin. That was fantastic. I really appreciate it. And for those of you who have more